Acids. We now Point 2 amino acids vary in their acid. Point 2 peptides and proteins. We now turn to polymers of amino acids, the peptides and proteins. Biologically occurring polypeptides range in size from small to very large, consisting of 2 or 3 to thousands of linked amino acid residues. Our focus is on the fundamental chemical properties of these polymers. Peptides are chains of amino acids. Two amino acid molecules can be covalently joined through a substituted amide linkage, termed a peptide bond, to yield a dipeptide. Such a linkage is formed by removal of the elements of water, dehydration, from the alpha carboxyl group of one amino acid and the alpha amino group of another, Fig. 313. Peptide bond formation is an example of a condensation reaction, a common class of reactions in living cells. Under standard biochemical conditions, the equilibrium for the reaction shown in Figure 3-13 favors the amino acids over the dipeptide. To make the reaction thermodynamically more favorable, the carboxyl group must be chemically modified or activated so that the hydroxyl group can be more readily eliminated. A chemical approach to this problem is outlined later in this chapter. The biological approach to peptide bond formation is a major topic of Chapter 27. Figure 3-13 formation of a peptide bond by condensation. Shaded in light red. Three amino acids can be joined by two peptide bonds to form a tripeptide. Similarly, 4 amino acids can be linked to form a tetrapeptide, 5 to form a pentapeptide, and so forth. When a few amino acids are joined in this fashion, the structure is called an oligopeptide. When many amino acids are joined, the product is called a polypeptide. Proteins may have thousands of amino acid residues. Although the terms protein and polypeptide are sometimes used interchangeably, molecules referred to as polypeptides generally have molecular weights below 10,000, and those called proteins have higher molecular weights. Figure 3-14 shows the structure of a pentapeptide. As already noted, an amino acid unit in a peptide is often called a residue, the part left over after losing the elements of water, a hydrogen atom from its amino group and the hydroxyl moiety from its carboxyl group. In a peptide, the amino acid residue at the end with a free alpha amino group is the amino terminal, or N. Terminal residue, the residue at the other end, which has a free carboxyl group, is the carboxyl terminal, C-terminal, residue. Key convention: when an amino acid sequence of a peptide, polypeptide, or protein is displayed, the amino terminal end is placed on the left, the carboxyl terminal end on the right. The sequence is read left to right, beginning with the amino terminal end. Figure 3-14. The pentapeptide cerylglysyltyrosylalanololysine, SER, GLY-TYR-ALA-LU, or SGYAL. Peptides are named beginning with the amino terminal residue, which by convention is placed at the left. The peptide bonds are shaded in light red, the R groups are in red. Although hydrolysis of a peptide bond is an exergonic reaction, it occurs only slowly because it has a high activation energy, P28. As a result, the peptide bonds in proteins are quite stable, with an average half-life, T1/2, of about 7 years under most intracellular conditions. Peptides can be distinguished by their ionization behavior. Peptides contain only one free alpha amino group and one free alpha carboxyl group, at opposite ends of the chain, Fig. 3-15. These groups ionize as they do in free amino acids. The alpha amino and alpha carboxyl groups of all non-terminal amino acids are covalently joined in the peptide bonds, which do not ionize, and thus do not contribute to the total acid-base behavior of peptides. However, the R groups of some amino acids can ionize, Table 3:1, and in a peptide these contribute to the overall acid-base properties of the molecule, Fig. 3-15. Thus the acid-base behavior of a peptide can be predicted from its free alpha amino and alpha carboxyl groups combined with the nature and number of its ionizable R groups. Figure 3-15. Table 3. Protein number. Cytochrome. Ribonucleotides, lysis, chymotrypsinogen, hemoglobin, human, serum, hexokinase, Apollo, glutamate, titin. Like free amino acids, peptides have characteristic titration curves and a characteristic isoelectric pH, pi, at which they do not move in an electric field. These properties are exploited in some of the techniques used to separate peptides and proteins, as we describe later in the chapter. We should emphasize that the pKa value for an ionizable R group can change somewhat when an amino acid becomes a residue in a peptide. The loss of charge in the alpha carboxyl and alpha amino groups, the interactions with other peptide R groups, and other environmental factors can affect the pKa. The pKa values for R groups listed in Table 3:1 can be a useful guide to the pH range in which a given group will ionize, but they cannot be strictly applied when an amino acid becomes part of a peptide. Biologically active peptides and polypeptides occur in a vast range of sizes and compositions. No generalizations can be made about the molecular weights of biologically active peptides and proteins in relation to their functions. Naturally occurring peptides range in length from 2 to many thousands of amino acid residues. Even the smallest peptides can have biologically important effects. Consider the commercially synthesized dipeptide L-aspartyl-L-phenylalanine methyl ester, the artificial sweetener better known as aspartame or NutraSweet. Many small peptides exert their effects at very low concentrations. For example, a number of vertebrate hormones, Chapter 23, are small peptides. These include oxytocin, 9 amino acid residues, which is secreted by the posterior pituitary gland and stimulates uterine contractions, and thyrotrophin-releasing factor, 3 residues, which is formed in the hypothalamus and stimulates the release of another hormone, thyrotrophin, from the anterior pituitary gland. Some extremely toxic mushroom poisons, such as amanitin, are also small peptides, as are many antibiotics. How long are the polypeptide chains in proteins? As Table 3:2 shows, lengths vary considerably. Human cytochrome C has 104 amino acid residues linked in a single chain. Bovine chymotrypsinogen has 245 residues. At the extreme is titin, a constituent of vertebrate muscle, which has nearly 27,000 amino acid residues and a molecular weight of about 3 million. The vast majority of naturally occurring proteins are much smaller than this, containing fewer than 2,000 amino acid residues. Some proteins consist of a single polypeptide chain, but others, called multi-subunit proteins, have 2 or more polypeptides associated. Non-covalently, Table 3:2. The individual polypeptide chains in a multi-subunit protein may be identical or different. If at least 2 are identical, the protein is said to be oligomeric, and the identical units, consisting of 1 or more polypeptide chains, are referred to as protomers. Hemoglobin, for example, has 4 polypeptide subunits, 2 identical alpha chains and 2 identical beta chains, all 4 held together by non-covalent interactions. Each alpha subunit is paired in an identical way with a beta subunit within the structure of this multi-subunit protein, so that hemoglobin can be considered either a tetramer of 4 polypeptide subunits or a dimer of alpha-beta protomers. A few proteins contain 2 or more polypeptide chains linked covalently. For example, the 2 polypeptide chains of insulin are linked by disulfide bonds. In such cases, the individual polypeptides are not considered subunits, but are commonly referred to simply as chains. The amino acid composition of proteins is also highly variable. The 20 common amino acids almost never occur in equal amounts in a protein. Some amino acids may occur only once or not at all in a given type of protein, others may occur in large numbers. Table 3:3 shows the amino acid composition of bovine cytochrome C and chymotrypsinogen, the inactive precursor of the digestive enzyme chymotrypsin. These 2 proteins, with very different functions, also differ significantly in the relative numbers of each kind of amino acid residue. We can calculate the approximate number of amino acid residues in a simple protein containing no other chemical constituents by dividing its molecular weight by 110. Although the average molecular weight of the 20 common amino acids is about 138, the smaller amino acids predominate in most proteins. If we take into account the proportions in which the various amino acids occur in an average protein, Table 3:1, the averages are determined by surveying the amino acid compositions of more than 1,000 different proteins. The average molecular weight of protein amino acids is nearer to 128. Because a molecule of water, Mr. 18, is removed to create each peptide bond, the average molecular weight of an amino acid residue in a protein is about 128. 18 is equal to 110. Table 3:3 amino acid composition of 2 proteins. Bovine cytochrome. See bovine chymotrypsinogen. Amino acid. Number of residues per molecule. Number of Some proteins contain chemical groups other than amino acids. Many proteins, for example the enzymes ribonuclease A and chymotrypsin, contain only amino acid residues and no other chemical constituents. These are considered simple proteins. However, some proteins contain permanently associated chemical components in addition to amino acids. These are called conjugated proteins. The non-amino acid part of a conjugated protein is usually called its prosthetic group. Conjugated proteins are classified on the basis of the chemical nature of their prosthetic groups. Table 3-4, for example, lipoproteins contain lipids, glycoproteins contain sugar groups, and metalloproteins contain a specific metal. Some proteins contain more than one prosthetic group. Usually the prosthetic group plays an important role in the protein's biological function. Table 3-4. Class lipoprotein, blood, glyco, phospho, hemopro, flavo, metallo, ferritin al. Summary 3.2 Peptides and proteins. Amino acids can be joined covalently through peptide bonds to form peptides and proteins. Cells generally contain thousands of different proteins, each with a different biological activity. Proteins can be very long polypeptide chains of 100 to several thousand amino acid residues. However, some naturally occurring peptides have only a few amino acid residues. Some proteins are composed of several non-covalently associated polypeptide chains, called subunits. Simple proteins yield only amino acids on hydrolysis. Conjugated proteins contain in addition some other component, such as a metal or organic prosthetic group.